Namaste students. I'm Jivin Dr. Aziz Mahmood Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, and Jyoti Vidyapith Women's University, Jaipur. Today, I'm going to discuss about the topic genetic recombination of heavy chain. In the previous video, we discussed about genes of the immunoglobulins, genetics, recombination of the light chain, and now genetic recombination of the heavy chain. So first we discuss about the genes of the heavy chains. Repeatedly we are discussing it. So in the heavy chains, if we talk about the heavy chains, there are some genes which make the genetic organization very diverse. So there are three genes there in the heavy chains. The first one is V genes, the second one is D genes, and the third one is J genes. V is for the variability, D is for the diversity, and J is for the joining. The variable region, V gene makes some variability, means different kinds of genes will make different kinds of the antibodies. D will make the diversity in the gene organization of the immunoglobulins, which makes the number of antibodies in large numbers. And the J region will combine the variable region to the constant region. So that is the use of the variable region in D region, the gene. So in the heavy chain, there are 50 genes, 50 V genes, and 20 D genes, and 6 J genes are present in the heavy chains. And what is their use? If we talk about the use of D genes, V genes, D genes, and the J genes, what is their actual functions? Suppose there are 3 portions. If there are 3 portions, only 3 portions will be counted, not all the 50, 20, and 6. 15 to 20 is equal to 1000, and 6 is equal to 6000, but the probability is more there than the quantity. Suppose that V1 is selected here, okay, D1 is selected here, and J1 is selected, so it will make one combination. Suppose V1 is selected, D2 is selected, and J1 is selected, it will make another combination. Suppose V1 is selected, D3 is selected, and J1 is selected, it will make 3 combination. Suppose V1 is selected, D4 is selected, and J1 is selected, so it will make one combination more. Suppose V1 is selected, D5 is selected, and J1 is selected, so it will make. So from one single combination we have prepared, so from one single V1 we have prepared 6 combination from one single V and one single J. We can also expand it when we select 6 more J. 6 into 6 is equal to 36. So from only V1 we can select a total of 36 combinations only from the V1. So it is possible that we can produce more and more combinations from a single type of gene. So that is the use of these VTJ recombination, because they will make some diversity, they will make diverse group of antibodies. So here we must understand how they will make the diversity. The recombination in the heavy chain is known as the VTJ rearrangement. Okay. And heavy chain synthesis use the same mechanism that was used for the light chain. In the early stage of the particular B cell, two rearrangement of the germ and DNA will occur. The first thing is the D segment alongside the 1J segment. The second brings the V segment next to the D unit, fixing the nature of the H cell. The rearranged heavy chain genes consist of the following sequences starting from the 5' end: the exon, intron, VDJ segment, intron, and a series of C gene segment. The rearranged DNA is then transcribed along the closest C region genes micro, mu, and the delta. So how the VDJ recombination is formed? Here first we describe here. This is the double stranded DNA where 50 V genes are there, 20 D genes are there, and 6 J genes are there, which are attached to the J region and the heavy chain, different heavy chains. If we talk about the DG joining, first the joining will be made between the D and the J. D and the J. Suppose here D1 is selected and J5 is selected. So there will be cut from D1 end and the D6 end. So the D2 and the D6, the D genes between the D2 and the D6 will be cleaved and they will be rejoined by the DNA ligase in there. After that, there will be a cut from the G1 and J5. So after a nuclease cut between the G1 and J5, the J1 and J2, J3, J4 and J5 will be removed from this DNA molecule and they will be again rejoined by the DNA ligase. So here D1 and the J5 will be here. So first in the DJ joining, D1 is joined here and a cross will be made here between the J1 and J5. This is the BTC joining. After that, there will be a cut from V1 to V1 to VT. Suppose here V1 is selected, sorry V2 is selected, so there will be a cut from the V1 and V2, and there will be a cut from the V2 and V50. So V3 to V50 will be cleared out here, and only V1 and the V2 will be there. After that, there will be cut from the V1 and the V2 and they will be removed here. So only V2 will be here, D1 will be here of the D chain and J5, J6 are here from which J6 will be cleared out here. Okay, J6 will be cleared out here. After that, the like heavy chain will be there, they will be joined together. Here one thing must be kept in mind that In the DJ combination, it was a DNA molecule which undergone the transcription, making a RNA molecule. And after this molecule formation, some post-transcription modification were made, which removed J6 and V2, D1, and J5 combined together with a primary RNA transcript of the uh, heavy chains. So this is the mature mRNA molecule, which next further undergoes DNA, sorry, translation, which will make a complete heavy chain molecule. So this is the mechanism of DNA rearrangement. Here, heptamer, monomer will be the two RSS molecule which are joined together between the transcript. Here, this RSS molecule. Okay, recombinant signal sequences are here. These are the recombination signal sequences which function in the recombination. So this enzyme bind on the on this region which need to be cleaved. If we talk about the rearrangement, so RSS genes bind here. Suppose if we need to remove V1, so it will bind here. Okay, this RSS molecule will bind here, and this molecule will make some cleavage here, which undergoes the cleavage of this V1 region and a cleavage between the V2 regions, which will undergo the cleavage of V1 region, and the V1 region will be removed. Suppose here the D1, suppose here the D1, D2, D3 are there, and for the removal of the D2, a molecule that is the RSS molecule will bind here, and it will make a cut here, and it will make another cut near the uh, D6. So it will bind at two places, and it will make two cuts which will remove the D2 region also. So this is the mechanism by which these cuts are made. Okay, so this is the basic. Here they bind on this way. Okay, if one turn are recombinant sequence are there, they will bind in the one turn in this pattern, and if there are two turn RSS molecules, they will bind in this manner here like this, and they will undergo the cleavage in this manner. One will cleave here, one RSS will cleave here, and the second RSS molecule, RSS gene will cleave here. This will undergo the complete cleavage of the DNA molecule. So this is the complete sequence. Now we talk about the class switching. Class switching means if we talk about preparation of a particular kind of antibody, for example, if we talk about the preparation of the immunoglobulin A, immunoglobulin G, immunoglobulin M, immunoglobulin D, so how they will be prepared. So this process is known as the class switching process. So it is completely dependent on the heavy chain, which are mu, gamma, alpha, or epsilon. So how the recombination of class switching is conducted? Suppose that we have undergone the VDJ recombination here. After VDJ recombination, suppose there is a requirement for the preparation of the immunoglobulin G. Then here a cut will be made here between the mu and the gamma 1. Okay, so this will be cleaved out here. Mu delta and gamma 3 will be cleaved here, and alpha 2 will be remain here. After that, alpha 2 will be removed here, then only gamma will be here. So the IgG1 will be prepared here. Suppose if we there is a requirement of the preparation of IgG3, so there will be a cut from mu to gamma 3, and then it will undergo the transcription, and a post transcription modification will be made here, which undergo the gamma 1 cleavage of the gamma 1 and the alpha 2. After that, the immunoglobulin will be IgG3. Suppose if there is a requirement of the preparation of the alpha 2, means immunoglobulin A, so there is a cleavage from mu to gamma 1. So this sequence mu, delta, gamma 3 and gamma will be cleaved, and the remaining alpha 2 will be found out here after the J resin, which will prepare a complete immunoglobulin A. So this is the class switching, which indicates that the next molecule which is after the J resin will be considered as the immunoglobulin class. So if suppose if there is a mu, there is not any kind of recombination, then it will prepare a mu. It will prepare immunoglobulin M. And if there is a requirement of a preparation of D, so the mu will be here, and immunoglobulin D will be prepared along with the immunoglobulin M. So this is the idea of the class switching. So it is complete here. Class switching, the process of the preparation of the immunoglobulin M, immunoglobulin D, immunoglobulin A, immunoglobulin E. So this is all about the preparation of immunoglobulin.